Okay, so thanks for having me. I'll talk a bit about decoders and give some examples from my PhD work in the Moser lab with some examples from the spatial navigation system of rats. So to start off, it's fair to say that one of the main goals of systems neuroscience is to be able to relate neural activity to something observable in the outside world, something like sensory stimuli or motor output or whatever we can measure. And when you record from one or only a few neurons, you're typically dealing with the encoding problem of how these observable variables explain the firing of single neurons. But now that we have these amazing technologies where you can record from hundreds of neurons, we can do the opposite task of decoding the stimuli or the external variables from the neural activity. And this is the process of decoding. And I'll give some examples from the system I'm working with, and in the process, talk about some considerations for when you do decoding analysis. So in our case, the observable variables are usually something navigation related. So we typically have a rat running around in an open box, and we track the animal's 2D XY position and its head direction. And then we record neural activity from the hippocampal formation, typically medial entorhinal cortex and neighboring areas with neuropixels probes. And if we start with the encoding or the tuning curves, we typically find cells that are tuned to position. So these grid cells that fire in these very regularly spaced locations in an environment, or these head direction cells. So these are polar tuning curves showing the firing rate of a neuron with respect to the animal's head direction in the environment. So this cell fires selectively when the rat is running east. And then if you record from enough of these neurons, we're able to perfectly decode whatever we want of these spatial variables. But sometimes we also get some surprises, and we see that even though we have hundreds of neurons, what we decode doesn't perfectly match what we track. And I'll try to argue that this might be interesting in itself and may reveal some hidden internal processes. And this is enabled by the high temporal resolution and the large scale of these recordings. So I'll now walk through an example from with head direction cells recorded in the dorsal precebiculum. So a large number of the cells in precebiculum are tuned to the animal's head direction. And here you see the spiking activity of 280 of such cells over a period of a minute. And to the right here, you see the animal's head direction during the same period. And our task now is to decode the head direction from this raw spiking activity. And a couple of things to note about this problem is that we're trying to decode a continuous one-dimensional circular variable. And this is quite well sampled in not so long time, and makes the problem a bit easier. So the first thing we have to do is to transform this raw spiking data into a spike count matrix. So we bin the spikes in time bins. Here it's 10 millisecond time bins with some smoothing. And then you end up with this neurons by time matrix. And we also need, at least for this approach, tuning curves for all the neurons. So here each row is the tuning curve of one head direction cell. And we can then decode head direction by essentially comparing the activity pattern at one moment in time with the expected activity pattern for each head direction. For example, by computing a simple correlation between these two columns, and then repeating the operation for all columns in both matrices, we end up with this matrix of correlation values, where you see that for each time bin, we have correlation values for each head direction bin. And it's typically one head direction bin that's mostly correlated with which yields the maximal correlation between the instantaneous activity and the expected activity for each head direction. And from this, it's quite simple to read out the represented head direction at any given time. We can also use Bayesian decoding, which works in a slightly different way, but the result is essentially the same. Then you end up with a posterior probability matrix instead, which sums to 1 for each time bin. And as you can see, again, you have this typically one head direction bin with the highest decoding probability for each time step. And then you can extract either its peak or some kind of weighted average to get an estimate of the decoded direction plotted in blue here. And as you can see, it matches the tracked head direction very well. And this is not surprising given that all of the neurons are strongly tuned to head direction. And in fact, we could have seen this by just sorting the spikes by the preferred heading of each neuron. You can essentially read out the population signal by eye. And now we'll move on to a few different other areas with head direction cells. So the head direction signal propagates from the anterior thalamus to the presubiculum and also to the parasubiculum. And if we look at the neurons in each of these areas, we see that they are all tuned to head direction. Here are 3 example neurons from each region. But you see that their properties varies a bit. So in the anterior thalamus, you see that the peak firing rates are very high compared to presubiculum, and in parasubiculum, you see that the tuning curves are quite broad. And this variability in firing rates has some implications for decoding. Like you don't need that many neurons if all of them have a very high firing rate, and also if your tuning is quite broad, you may not be able to decode as precisely as with sharper neurons. And by recording from each of these areas simultaneously with the multiple chronically implanted neuropixels probes, we can first just by eye in these sorted raster plots see how these population signals evolve in each of the 3 regions. And what we can also do is to apply this same decoding approach. And what I've plotted here is the offset between the decoded direction and the animal's tracked head direction. And you can see that in both anterior thalamus and presubiculum, you see that the decoded direction overlaps very well with the tracked head direction. But in the parasubiculum, you see this peculiar offset to either side. So if we had just looked at the tuning curves, we may have said that, okay, these neurons are just noisy, maybe they're just noisy. But when we see this bimodal distribution in the decoded direction, it could be that these neurons are actually coding for something else. And we'll take a closer look at these parasubiculum cells again. Here's the animal's tracked head direction over a few tenths of seconds, and the spikes from hundreds of these parasubiculum cells. Then if we zoom in on a small, shorter segment and look at the spiking activity at the timescale of theta cycles, so each theta cycle lasts about 125 milliseconds and are indicated with these shaded rectangles. You will see these packets of coordinated spiking activity to either side of the animal's head direction. And if we apply our decoder, we will see that the decoded direction also alternates back and forth to either side of the head direction. And again, if we plot the offset between tracked head direction and decoded direction over the whole session, we see this striking bimodal distribution, indicating that these cells are not actually coding for head direction, but some other signal that alternates from side to side. And since we couldn't really find an external correlate that matched these dynamics, we named this signal the internal direction signal. And again, these cells appear as broadly tuned to head direction when you look at their conventional tuning curves. But if you recompute their tuning with respect to this decoded signal, you see that they're actually quite sharply tuned to this signal, again indicating that this is what these cells are doing. So by applying this decoding analysis, we think we found out what these cells are actually doing. And we'll now move on to these spatially tuned cells that are the grid cells in the medial entorhinal cortex that have very clear position tuning. We can record activity from a lot of these cells at the same time. And again, apply the same decoding approach where we correlate instantaneous population vectors with the expected activity pattern for each position in the environment to get a decoded position. And if we do this and plot the decoded position over time, indicated by this colored blob, we see that the decoded position starts around the animal's current location and then sweeps outwards in these stereotyped trajectories within each theta cycle. So this video is slowed down. And what we found is that these trajectories to the left and right are actually coordinated with a decoded signal from internal direction cells. So what we take from this is that these grid cells are actually not just coding for the animal's position, but also these locations around in the environment. And lastly, we can take this a bit further and apply our decoder across behaviors to see what this signal is and what it reflects. So here in a task where the animal is chasing this little piece of food that is dragged around in the open arena. And if we apply our decoder to the internal direction cells, we see that these cells seem to indicate the direction towards the piece of food while it swings behind the animal's back, for example, and perhaps reflecting where the animal is directing its attention. And by doing this again across regions, we can see that there's a functional split in the population signals across areas. And we can also do this during other behaviors such as backwards walking. And lastly, because we can use our knowledge about the spatial navigation system and the topology of the population activity in grid cells, which we know are constrained to this toroidal manifold, to apply our decoder during sleep. So we of course can't compute tuning curves when the animal is sleeping and not moving, but we can, because the manifold persists, we can use tuning curves from wake and neural activity from sleep to decode the represented position during sleep. And first we see an example from how this for how this decoder works during wake. So we decode position from two grid modules simultaneously decoded, and can plot the trajectory on the torus unwrapped into physical space for both of these modules as an animal runs around in the box, and do the same during a minute of REM sleep. And what you see is that the decoded position from these two models play out an extended trajectory of several meters that is coordinated across modules. And again, if we zoom in on these longer trajectories, we see shorter sweeps playing out on the finer timescale. And with that, I think it's time to wrap up, and hopefully I've shown some of the applications of this decoding approach, and some of the considerations you need to pay attention to when you do this analysis. Um, and I would like to thank everyone in the Moser lab where this work was carried out, including my supervisors and the collaborators who participated in this work. Thank you.